And so good morning to everyone joining us. I'm excited to introduce Samsung Biologics' newly launched organoid service, Samsung Organoids. So I'll begin today's presentation with a brief overview of Samsung Biologics' core capabilities from our development to manufacturing services prior to moving into Samsung Organoids. So Samsung Group is a strong, stable, and sound partner. The company was founded in 1938, and within the 85 plus years since its inception, has made successful contributions across a broad range of diverse industries, including electronics, engineering, and finance. Samsung Group is ranked fifth globally with a $100 billion brand value and over $243 billion market cap. Samsung Biologics is the healthcare arm of Samsung Group. It was founded in 2011 and has served for the past 14 years as a leader in the CDMO space, having successfully supported programs for large pharma and biotech clients globally. And as a whole, Samsung Group employs over 500,000 employees in 91 countries, with Samsung Biologics employing approximately 4,500 of those. So Samsung Biologics' portfolio supports the development and manufacturing of a broad range of modalities. So we can support the manufacturing of monoclonal antibodies, complex multispecifics, antibody-drug conjugates, recombinant proteins, and mRNA and Fc fusion proteins. The bottom panel shows our locations and capacity. From the left, BioCampus 1 houses our plants 1 through 4. All of the plants and locations are located in South Korea. Plant 5 was introduced or opened earlier this year and marked the start of our BioCampus 2 expansion. This expansion is expected to complete in 2032. From the center, the ADC plant is where all of our organoid work is being performed. This ADC plant is a GMP-aligned facility, and it's actually located 1 block away from BioCampus 1. Now, we maintain a global reach to our clients through our East and West Coast United States sales offices, and we recently just opened a new office in Japan, Tokyo, this year. So we are continuously investing in bringing on new modalities to support our clients and new services throughout the drug development journey. So what initially started as the launch of drug substance and drug product manufacturing services over time and based on client need has evolved into us bringing on development services, mRNA, and ADC services. And now with the launch of Samsung Organoids, we can support our clients at the early preclinical research and discovery stages. Now, our business model is a 3-pronged business model. So this slide just provides a high-level overview of that. Our organoid service leverages patient-derived models for efficacy testing, drug screening, and we can also support immune co-cultures with biomarker ID available. For our development services, these are built to support clients in late discovery to IND and beyond with customizable CMC solutions available. On the manufacturing side, we provide flexible drug substance support ranging from 1,000 to 15,000 liters for commercial supply. And for drug product, we support vial, lyophilized products, PFS, so we're bringing pre-filled syringes on shortly, as well as ADC-DP. Now, all of our services are performed within our centralized labs that are equipped with regulatory support. So here, our development services, they're typically from late discovery to IND and beyond. What we can do within these services is provide accelerated development timelines. We support and customize these services for large pharma as well as biotech clients, and we work with a large number of biotech clients utilizing this service. So what we can potentially do is using accelerated timelines beginning at the late discovery stage, move in and continue through cell line development to GMP manufacturing. We also offer select tailor customizable CMC solutions. These can be optimized based on the characteristics of your drug as well as the developmental stage of your program. Now, we pride ourselves on being a flexible partner with the capacity to fit any strategy. Our current drug substance capacity is 784,000 liters. And with the recent BioCampus 2 expansion that I described earlier and is shown below, our DS capacity is set to nearly double to 1.3 million liters by 2032. For drug products, we can fill approximately 20,000 vials per day with lines that support liquid and lyophilized products. And by October 2027, we will be launching CGMP-ready pre-filled syringes. And so taken together, our capacity permits us to fully support your programs from early clinical supply through to large-scale launches. So if you're interested in our CDO or CMO services, I am happy to connect you with the relevant teams to begin discussions on how we can support your programs. I'll now move on to Samsung Organoids in detail. So the challenge in drug development is that it is a long and costly process. It often takes more than a decade and billions of dollars before a candidate drug reaches human trials. And at that point, approximately 10% of those drugs will ultimately make it into the clinic. Now, this high attrition rate is driven in part by the limited predictive value of preclinical models, and so namely, animal models and 2D cell lines. In contrast, organoids provide a human-relevant model system that enables our clients to screen compounds early in an effort to reduce the attrition rate before they reach clinical trial. And so if you integrate this screening process early into your program, you can build a more efficient development pathway with shorter timelines and ultimately reduced risk. Now, importantly, the launch of this platform is coming at a time where the FDA is actively recognizing and encouraging the use of new approach methodologies. And this is including organoids, microphysiological systems, and computational models. And the goal of this push is really to reduce the reliance upon animal testing, but also to advance models that better reflect human biology. And so this shift signals that regulatory agencies are really ready to integrate these approaches into drug development as they recognize their value in productivity and reducing risk. And so for our partners, there's never been a better time to incorporate organoid-based data into your preclinical screening because the regulatory environment is currently moving in the same direction. Now, our current platform is focused on establishing patient-derived organoids directly from solid tumor biopsies. Patient biopsies are collected at our clinical collaborator sites at the time of surgery, dissociated ex vivo, and then cultured into 3D organoid models. Afterwards, these models are undergoing a rigorous QC process and characterization before being added to our continuously growing biobank for commercial use. For client studies, we utilize these organoid models at very low passage, and that's in an effort to maintain the heterogeneity and molecular fidelity of the original patient tumor. And so by screening your compounds across our biobank, we provide data with higher clinical relevance across a broad range of solid tumor types. And as we are actively expanding and growing this bank, we are looking into opportunities to expand into additional disease areas and normal tissue indications for safety and toxicity testing. And so our biobank will continue to grow over time. Now, this slide is really illustrating how we are supporting our partners across the early research and discovery stage through late stage CDO to manufacturing. The organoid platform provides a method for clients to perform drug screening to identify lead drugs or candidates that they would like to fast track into clinical trials and into development. And this is utilizing models that are clinically relevant and supported by NGS sequence data. And so utilizing that NGS sequence data and the additional patient treatment information and multimodal data sets that we provide on each organoid, we can help aid your preclinical development and assist in identifying biomarkers of response or resistance. And so by combining all of these screening results, the sequence data and patient treatment information, insights that are gained through this process early on can serve as a foundation for guiding clinical trial patient selection, as well as expanding treatment indications. And so this is basically, in short, us providing a method where you can test your agents, identify your leads, and then move on to development and manufacturing very seamlessly. Now, once a client has screened and identified their asset using Samsung Organoids, we can, the next natural step here is really to move on to support developability assessment of your molecules using our DevelopIC service prior to continuing your journey through cell line development and onwards to manufacturing. And so that means that your most promising molecules are evaluated not only for their biological relevance, but also for their development readiness. This slide is just highlighting the depth of clinical data that I mentioned earlier that we are providing for each organoid model being tested. These are divided into 7 categories, which are focused on patient demographic information, diagnosis, treatment history and results, as well as immuno-oncology diagnostic data and molecular pathology. And this is data that we can share where we've obtained it from our clinical collaborator site for each individual patient. Now, this slide provides a broad overview of our services at a high level. And as I mentioned earlier, our organoids are established directly from patient tumor biopsies collected at the time of surgery. And so once the organoids are established, they undergo QC before being commercially available. And when I mention that, what I'm describing are the QC metrics that are shown on this slide. And so what we will do is we will perform assays to measure growth characteristics. We will run DNA and RNA sequencing of those organoids, look at histological validation just to confirm the cancer type is appropriate with the tumor type that the biopsy was from the biopsy that was utilized to establish the organoid. And we also perform mycoplasma testing. And only models that pass all of these checks can advance into screening. From there, we support a wide range of assay formats. Our standard assay is an 8-day organoid monoculture assay. These are run typically in 384 well plates where we measure cell viability. This is typically by performing an 8-point dose response of the client's test agent with positive and negative controls. We also offer advanced co-culture assays. These are for clients that want to model the tumor immune microenvironment utilizing organoids with immune cells. These are typically run in 96 well plates. What we can do is we can source allogenic PBMCs from external vendors, or we can utilize the autologous PBMCs or fibroblasts that we are actively deriving from these patient tumors. And these are contained within our biobank. And so we're getting to the point where we can really nicely model the tumor microenvironment using cells derived directly from that patient. We also support immune cell therapies. And so if anyone is establishing a CAR-NK or a CAR-T cell that they are using or developing in-house, we can certainly support an immune co-culture utilizing those cells as well. Now, the readouts typically for these co-culture studies are cytotoxicity assays, so LDH assays, high content imaging, flow cytometry, and cytokine detection via ELISA or CBA assay. What's most important here, and I'd like to kind of bring up, is that we offer rapid turnaround times with approximate 5-week timeline from project initiation to results. And this is all supported by flexible study designs, high quality data output, and reproducible findings. And so when considering organoid partners to work with, why choose Samsung? First, we have a track record of providing high quality data and service reliability. As I mentioned, all the organoid work is being performed in our GMP aligned ADC facility with standardized workflows and very traceable lab processes. We also secondarily pride ourselves on our flexibility by permitting clients to custom design their studies for specific objectives. And so there's no limit as to how you design your study, as to how many organoid models you're testing, and what types of readouts you'd like to utilize to measure responses. And lastly, we also engage in custom model building to support your programs. And so what we would be able to do is go out and source patient tumors from rare cancer types or from pretreated patient populations in order to build a model set that really reflects the patient population that you're targeting. And so Samsung is growing into the three verticals shown, which are capacity, geography, and portfolio. I mentioned that geographically, we expanded by opening a new sales office in Tokyo, Japan. Portfolio-wise, we've launched Samsung Organoids and are also investing in next-generation ADCs and adenovirus. We have also expanded our technology and platform for our CDO business and included AI and digital transformation throughout our workflow and processes. Capacity-wise, I mentioned that we recently opened or launched BioCampus 2 with Plant 5, and that's expected to be completed in 2032. As far as the ADC facility that was completed earlier this year, we will be introducing or finalizing conjugation and fill finish by Q1 2027. And so thank you all for your time. We have a number of groups that are exhibiting currently at CPHI, and the Organoids team will be at World ADC in November. So if you're interested in connecting with us and further exploring how we can support your programs, please feel free to reach out, and we can arrange for that. So thank you. And Perry, I'm happy to take any questions that may have come up during the presentation.
Thank you, Alexis. Very, very exciting stuff. Really interesting. You talked about the clinical relevancy of the data using organoids. What kind of impact does it have on risk when it's compared to animal models and cell assay?
Sure. So I think what's important is that traditional animal models, they remain important in the drug development process. But the value in utilizing organoids for screening is that they capture patient complexity much earlier. And so that means that what you're doing is you are generating human relevant data early before investing heavily in lengthy animal research or PDX studies, for instance, as well as getting to clinical work. And so this reduces the risk of late stage failures.
We have a two-part question. Are patient organoid tumor samples being taken from U.S. populations? And then are there any future plans for facilities in the U.S.?
Sure. In terms of the first question, yes, we are sourcing organoids from multiple different clinical collaborator sites. Currently, the bank that we have established is utilizing organoids derived from our clinical collaborator. So it's the Samsung Medical Center. So this will continue to grow, and we're expanding on that. Secondarily, Samsung is always looking into expanding. And so, of course, they've been looking at the potential to expand into the US as well as other markets.
Okay, so we'll take that second part as perhaps. So you mentioned a 5-week turnaround. How significant is that? And how does that support key decision making?
Sure. So in large molecule development, timelines are long. The advantage of a 5-week turnaround is it's about saving time, but also about just enabling a faster go, no-go decision. And so this goes back to the attrition that I described earlier, where drugs are carried all the way through from discovery to clinical trial, and then they fail. And so by utilizing these human relevant organoid model systems earlier, what you're doing is you're cutting out those molecules that will not work or do not work in patient samples that are presumably primary enough, so that clients can focus their efforts on promoting candidates that would be available to elicit a response in patients.
Yeah, interesting. Okay. Thanks for explaining that a little more. Bioinformatics support. Can you talk a little bit more about that and the support you offer in identifying biomarkers?
Sure. So we actually have a couple case studies where we do this. And I mentioned early that we are characterizing our organoid models by DNA and RNA sequencing. And so when a client runs a drug screen and they identify drugs that respond or models that respond and don't respond to their specific drugs, we can support biomarker ID to identify biomarkers of response or resistance to their therapies. We can also integrate patient data into that, and so that can be useful for customizing their analyses to provide actionable insights into how they design their clinical trials, or if they want to expand in a specific patient population where the drug, worked but wasn't necessarily expected as they designed the organoid system that they were going to test their drug in.